A very warm welcome to all the listeners of the MedSynapse OncoShorts podcast series. OncoShorts podcast focuses on the latest clinical and research developments in the field of oncology. Today's podcast episode focuses on the trial, titled Phase 1 Open-Label Trial of Intravenous Administration of MVA-BN-Brachyury TRICOM Vaccine in Patients with Advanced Cancer. Brachyury is a transcription factor that is overexpressed in many solid tumors, such as lung, breast, ovarian, chordoma, prostate, colorectal, and pancreatic adenocarcinoma. It is a member of T-box family of transcription factors and characterized by a highly conserved DNA binding domain. This transcription factor has been associated with treatment resistance, epithelial to mesenchymal transition, and metastatic potential. Till date, 5 investigational therapeutic cancer vaccines targeting brachyury have been studied. One such vaccine is a recombinant vector-based therapeutic cancer vaccine, named Modified Vaccinia Ankara or MVA. The Bavarian Nordic or BN-Brachyury therapeutic cancer vaccine is currently under investigation in a Phase 2 study, in combination with radiation therapy for chordoma. In this trial, the results of first-in-human clinical trial of an intravenously administered therapeutic cancer vaccine targeting brachyury has been described. The investigational biologic product, known as MVA-BN-Brachyury, is a liquid-frozen, highly attenuated, live recombinant virus. The primary objectives of this trial were to assess the safety and tolerability of the vaccine and to determine the recommended Phase 2 doses. Patients with non-small cell lung, small cell lung, breast, ovarian, prostate, colorectal, pancreas, hepatocellular, bladder, and kidney carcinomas, with histological evidence of overexpression of brachyury, were included in this trial. 3 different doses of vaccine were explored, that is 1 × 10⁻⁷, 1 × 10⁻⁸, and 1 × 10⁻⁹ infectious units. Concern for potential cytokine release syndrome, that is CRS, was the rationale for proceeding with escalating Phase 1 dosage studies. A total of 13 patients were treated between January 2020 to March 2021. The vaccine was administered intravenously every 3 weeks with 3 administrations in total. Drug pharmacokinetics and immune activation studies, along with the imaging studies at baseline, 1 and 3 months post-treatment were done. The primary endpoint was safety and tolerability, as determined by the frequency of dose-limiting toxicities. Secondary endpoint included determination of the recommended Phase 2 dose. From this trial, it was observed that the overall cohort included 10 patients, that is 76.9% with chordoma, 7.7% with small cell breast cancer, 7% with colorectal cancer, and 7.7% with prostate cancer. In this trial, no dose-limiting toxicities were observed, and no serious adverse events were attributed to the vaccine. Cytokine release syndrome, up to grade 2, was observed with no adverse outcomes. A dose effect trend was observed for fever, chills or rigor, and hypotension. 3 patients with stable disease experienced clinical benefit in the form of improvement in pain and decreased use of analgesics. Immune correlatives showed T-cell activation against brachyury and other tumor-associated cascade antigens. In terms of efficacy analysis, 1 patient showed partial response, 4 showed stable disease, whereas 8 patients showed disease progression. From this study, it can be noted that hospitalizations may be low in future studies, given the safety of intravenously administered MVA-BN-Brachyury TRICOM, and the low grade of CRS observed. Future efficacy trials may consider excluding patients with clival lesions due to potential for increased intracranial pressure from vaccine-mediated inflammation. Given the rarity of chordoma and the paucity of patients available and eligible to enroll on study, careful consideration of study design is of paramount importance. With this, we conclude today's OncoShorts podcast. Thank you for listening and being part of the largest community of doctors.